But now that you have these different cell stages, or this path that you delineated using the pseudotime, the final step is to find genes that are differentiated in the middle of this process. And for that, one of the packages, for example, in R, is doing TradeSeq. For PAGA, they also have a test that is kind of similar to this as well. So I'll just refer to this one, but the idea is quite similar. So what you need to do first is to find a curve in all the different conditions that you have. So here you have, for example, in orange, one of the trajectories. Imagine not like a bifurcation. And you have these cells here that will not express any of this marker. And after differentiation, another branch will express that marker. So then you fit a line through those. And then you can do clustering of the different genes. So for example, a cluster of genes, that are many genes that sustent this branch and other genes that do not. And the same thing can be done in other gene paths, as in here. So differential gene expression in trajectory is much more complicated than the normal differential expression, because you have many questions. One question, for example, which genes are conserved in the start of the trajectory, but are differentially expressed in the end of the trajectory in both trajectories? Or you want to find genes that are differentially expressed in the end of this trajectory and in the end of this trajectory. So there are many relevant questions that you can ask for it. And for all the different questions, you need to think about the different tests. So there are, in this package, for example, there are 5 different tests that you can use. So association test is to test if that gene is changing over time. So another test is start versus end. So I want to see if a gene is which is expressed in the end is different from the beginning. And this is within one single lineage, or like one single line, like this one. So for example, for this one, this gray orange one will not be differentially expressed, for example. Now between the two lines, as you can see, for example, for this lineage here is marked as DE because the end and the start are different. But now, for example, imagine these two lines, or like this one. So this one is not differentially expressed in the start or the end because they are the same, basically. But the difference between the ends between those two are differentially expressed. Now the pattern, or like the gene expression, is completely different, or the way the gene moves over time is different. Is it this one? It is. But for this one, they have exactly the same expression trend. It goes down and then it goes up. While in this one remains flat and the other one goes up. So it becomes way more complicated to find patterns and to answer your questions in this way. The same thing goes for early DE test, to see if, like, in the beginning, the gene expression is already different between the two lineages. And in the tutorials, you will see more about those differential tests and how to run each one of them. But just keep in mind that is a little bit more complicated than normal differential gene expression, and really depends on your biological question or on the curves that you find in your data.
And also here, I should mention that it's again becomes super important with a lot of cells, because what they do is a linear fit of the gene expression. So also these very lowly expressed genes with just a few cells here and there, they will still do a linear fit to, and then do this kind of test and call it something. So really plot those genes and look at them, not only the lines, but at the actual expression, because you get a lot of false positives with these methods.
Yeah.